Hey guys, this is our content video looking at Chapter 11, Determining Relatedness Between Species. So as a lead on from Chapter 10, where we started looking at evidence for evolution, this is similar. It is evidence for evolution, but it is a more accurate, more reliable way of determining how related two species are. Okay, so Chapter 11 focuses on this lot of key knowledge. Looking at molecular homology as evidence for relatedness between species, including DNA and amino acid sequencing, mitochondrial DNA, and DNA hybridization as a technique. We'll then look at phylogenetic trees and how they're used to demonstrate how related two species or a group of species are. And then we move on to look at an example of master genes, being the BMP4 gene, and how that has influenced the evolution of features in the Galapagos finches and cichlid fish in Africa. Right, so for starters, molecular homology, don't be scared off by the term. We looked in the last chapter at structural homology. Homology just being a comparison of how similar two things are. Structural, meaning that we're looking at how alike they are structurally or physically. Molecular homology is looking at how alike their molecules are, so comparing proteins that are found in particular species or a DNA sequence, a particular gene, to see how alike those two molecules are. And that gives us a bit more of a quantified number to establish how related they are. Definitely more preferred than structural homology. It's more accurate than comparative morphology is the other term that we would give that technique to look at how physically similar species are. And this diagram demonstrates why. So on the left side, we can see the setup that we would have of a phylogenetic tree based on how similar the species appear. So how structurally similar they are, comparing their morphology and how they look. These are the like groups that would be established. However, when we look at their molecular structures, so their DNA, and compare how alike they are genetically, we get a little bit of a different picture painted. So structures can appear similar on the outside, but then when you look beneath the surface, they aren't. They are a little dissimilar. So kind of like when we looked at the comparison between what the limb structure looks like on the outside compared to what the bone structure looks like underneath. So a similar sort of scenario. But we are looking even deeper at their molecular structures now and seeing that there are similarities between them that we don't see on the surface physically. And there are mutations that we can look at on a molecular level can occur in DNA, and related to that we know we can look at amino acid sequences, and mitochondrial DNA can also offer some differences between species. Okay, so looking at DNA comparisons first, we can establish how alike two species are if we look at their DNA sequence, and there's a particular sequencing technique that I'll talk about in a minute. But if we want an approximate idea of how alike two species are, we can use something called DNA hybridization. So that's where we take the DNA samples of a group of species that we want to compare. They are denatured and separated so that they become single-stranded. So if we heat DNA to a high enough temperature, the double helix will come apart and we'll have single-stranded DNA. We can cool that mixture down so that we have sequences line up with each other, and sometimes we'll get that hybrid DNA form. So one strand of species A has now joined with a strand of species C in this diagram. and formed some hybrid DNA. We can see similarly we've got species A with species B. We've got another hybrid strand here. That's not the end goal. That's not the final piece of the puzzle. I think I might just skip over to your textbook quickly and show you a diagram in the textbook. It does give the details of this quite well. Give me a minute while I find it. There we go. Okay, so this diagram in your textbook takes you through those stages quite nicely. But once we've got our hybrid DNA form, don't forget that there is this final strand of reheating the DNA, and that allows us to establish how alike the two species were. So if we've got our three examples here, this form of hybridization is tightly bound, so it's going to require a higher temperature in order to denature it. So we can assume that these two sequences are very similar. If we have partial hybridization, that's not going to require as high a level of heat to denature it that second time around. So this would be the highest temperature. This would be the next highest temperature required to denature it again. If there's no hybridization, then obviously it's going to be very little hybridization. There's going to be very little temperature that's going to be required to denature that sample. So that's how they establish how alike the two strands are that have hybridized. Okay, so that's DNA hybridization. If we want an exact method of comparison between DNA, then we're going to need to use DNA sequencing. It's unlikely that in an exam you'd be asked to recall the steps involved in a particular method of DNA sequencing because they are very complicated. But one method that's used is, I think that lots of people pronounce this as Sanger, the Sanger method. I like to say it's the Sanger method. So the Sanger method of DNA sequencing involves a technique called PCR and gel electrophoresis. These are two techniques that we're going to go through in better detail when we get into chapters 13 and 14 a little bit later. And you might look back on this then in your revision and it will make a little bit more sense. But essentially, we are building increasing sizes of DNA to partner with this strand that we want to sequence. And the final base in each sequence is fluorescent. So when it gets to the end of this short sequence, it will glow a particular color. If we then run that on using a DNA technique that allows us to separate DNA based on its size, we can get an order or a pattern of those DNA sequences, and it can be read off in that manner. So we can end up getting our final strand that was to be sequenced, because remember, we can't see the DNA and they don't actually have little C's and T's and G's. We need a method that we can actually visualize it, and the Sanger method is one of them. In your OneNote, you've got a link to an interactive that will help you maybe understand how the Sanger method works, and you can give your own little short description there. Okay, so that's DNA comparisons. We can also use amino acids to compare between species. So think of a protein that is found in our two species, or a protein that's found commonly across life. For example, DNA polymerase. We can't have DNA form without this enzyme DNA polymerase. So that is one protein that is found across all species. It has changed slightly from species to species. The same for RNA polymerase. But these common proteins, if they're found in two or more species, then you can use its sequence to compare how alike that structure still is. And if it's very similar, we can assume they are closely related. If it's accumulated more mutations, then we assume that they've been diverged, or the common ancestor for those species existed a longer time ago. So amino acid comparisons are not as accurate as DNA comparisons. Why? So have a think about why you think that might be. Why would it be better if we've got access to a DNA sequence? Why would it be better to compare the DNA sequence for the protein rather than the amino acids? And hopefully you said something along the lines of you see more differences. There are differences that can be found in DNA that would not show through in an amino acid sequence, silent mutations. So if there's a silent mutation that exists in the DNA, it would unfortunately, for comparison's sake, still give us the same amino acid. So even though you see the same amino acid when you're comparing the two protein structures, there may be a difference in the DNA that you're not seeing. But amino acid comparisons can be done. So there's an example here of a human compared to a mouse, rat, cow, and chimp. And the differences have been highlighted for us so we can see which are the closer related species. And you can sort of see that the human and the chimp have got more amino acids in common than the rest of the group. So humans' closest relative within this group would be the chimp. But we've got this color coordination going on here just to explain that they are ranked, the differences or the mutations are ranked as conservative, non-conservative, and semi-conservative. To understand what they mean, you've got to remember something about amino acid structure. So again, I'll just come back to your textbook and show you a diagram that demonstrates this. Ooh, all the way back to the start of your textbook. Bear with me while I find it. So when we're comparing amino acid structures, some amino acids are more alike than others. And that refers to their R group structure. So those R groups, remember each amino acid has a constant region, there we go, a constant region and a variable group, that R group. And this diagram in your textbook shows how the R groups of some amino acids are similar. So they're grouped together. So these R groups give the amino acids an acidic property. These R groups give the amino acids a hydrophobic property, and so on. So when we're looking at a conservative mutation, that means that the mutation here has not had a dramatic effect on the activity of the protein. The two amino acids behave in a similar way, and so they're conservative. It's conserving the action of that protein. Semi-conservative is sort of partway in between the two. It's close, but not quite. Non-conservative is where you've got an amino acid that is very different in its properties to the original amino acid, or the two amino acids that are at play are very different to each other. We would see more conservative mutations in amino acid comparisons because that if it's conservative and the R group is behaving in a very similar way, that allows the enzyme to retain its function. Non-conservative mutations are more likely to be detrimental and not have as much activity as the enzyme should. Okay, this idea of a molecular clock. So we've talked about mutations, and this idea is that we can quantify these mutations at a constant rate. So the molecular clock refers to the idea that if we can establish how many differences there are between two species in their DNA or in their amino acids, we can put a time to that. So it assumes that mutations accumulate at a constant rate, which we know they don't, And so it's not a very accurate measurement, but it's the best we've got. So if we assume that mutations accumulate at a constant rate over a given time, and we can see how many mutations difference there are between two species, we can give an estimate of how long ago those two species diverged. Mitochondrial DNA seems to be the only place I've got it in the PowerPoint, so I better talk about it now. Mitochondrial DNA is commonly used as a molecular clock because it accumulates mutations at a faster rate than nuclear DNA. The reason being is that mitochondrial DNA doesn't have the repair mechanisms that nuclear DNA does, so mutations often slip through and are accumulated faster. The theory that is covered by this course and in your textbook is that mitochondrial DNA is inherited maternally, and so it's passed on from mother to child, and so that we should be able to track maternal lineage back for all the generations. But recent studies challenge that idea. So I'm saying it's likely not to be examinable anymore. However, should you see something written in the exam about mitochondrial DNA being maternally inherited, just know that yeah, the key knowledge does tell us that it is still maternally inherited, and that is a fairly recent discovery that it's in fact not some of the time. Okay, phylogenetic trees. So once we've got this information, we know how alike two species are, we can use that information to build a phylogenetic tree to show us the relationship between that group of species. So you've got the example here from your textbook for flowering plants. The thing that will stand out most quickly when you are looking at, in this case, a DNA comparison, and I know it's a DNA comparison because I'm only looking at 4 letters, C, T, A, and G. But the thing that stands out most quickly, I think, is the outgroup. So an outgroup of a phylogenetic tree is the one that is the least similar. So when you can identify which one has the least in common with the other species, that is placed on the phylogenetic tree as an outgroup. So we always start with the root and a first common ancestor, a point of divergence. So at one point there would have existed a population of flowering plants that is the common ancestor of all of these current day flowering plants. And the first species or the first speciation event that took place has given rise to what has now become the pinus, or the pine tree. And then they kind of follow on from there. So the one that is second least alike is the corymbia or the eucalypt. So that one has diverged away next, leaving the final two, glycine and vigna, being the most closely related or the most recently diverged species. So that's an example of a phylogenetic tree being set up from some genetic information. These are much more complex phylogenetic trees that are shown here. If you're asked to draw a phylogenetic tree, it is most likely going to be a group of 4 to 6 at the most, I would think, species, and definitely not this many or this complicated. But you would have to be able to recognize the structures within these more complicated phylogenetic trees and be able to interpret and understand them. So the parts of a phylogenetic tree to pay attention to, some I've already mentioned, but I will just run through them. So we always start with an ancestral lineage, where the most ancient common ancestor of all of our species in question would be drawn. And this node here represents the common ancestor of all of our present day species that are just labeled here as taxon A to G. Just a side note, if, say, taxon G was drawn further away back here and not in line with the others, that is a sign that taxon G has actually gone extinct. So everything in line at this point is meant to represent present day. If one of those species is drawn further back, it represents an extinct species. But there's none of those in this example. And so then each node goes on and divides each time there has been a speciation event. Because it's a phylogenetic tree, the tree sort of naming continues, and we have branches to represent the final species that have emerged. And a leaf is where we place the label. A clade is the word used to refer to a group of similar species. So the green clade here represents a clade of taxon A, B, and C. That is a group of related species. This would also be a clade here in orange. However, I couldn't extend the green one to include just taxon D because I'm then ignoring some other related species. I could draw a bigger one around all of A, B, C, D, E, and F, and that would be a larger clade. But that's what that word refers to. The shorter the branch length indicates the fewer differences between species. So the most closely related species in this table, or in this tree, sorry, would be taxon B and C. But a group of related species we can refer to as sister taxa. And then the last one, which I did mention earlier, is that group that diverged first, that is least related to the rest of our species in question, and that is the outgroup. And then finally, we'll just quick look at master regulatory genes. So we have looked at regulatory genes before when we've looked at the lac operon, and that was sort of small scale. So we've looked at the fact that we can have a gene that regulates the action or the activity of another gene, being a regulatory gene. A master regulatory gene is sort of similar, however on a larger scale. So master regulatory genes don't just influence the expression of one gene, they influence the expression of many genes. So that's a good diagram there in your OneNote to represent that activity. So we have our one gene X being the master regulatory gene, and its protein that's produced has an action or it is a transcription factor to influence the expression or no expression of several genes that are present within that organism. And these genes are very important and highly used during development. So developmental genetics is a whole area of study all of its own that looks at these genes and how they control the growth of particular areas of an organism, in this case a fly. A gene would be expressed in this region here that will then influence the development of all of these features by controlling multiple genes. So the example of a master regulatory gene that we're asked to know about is this one called BMP4. You definitely need to have a read through the textbook on BMP4 and what information is there, because it's a complicated example and it's a hard one to understand. But BMP4 is a gene that controls the expression of a protein called bone morphogenetic protein 4. And it is the best explanation as to why we have seen some genetic phenomena kind of happen in two examples, the examples of the Galapagos finches and the cichlid fish. So here's where you want to have a look at the videos explaining what the physical differences are between the finch and the cichlid fish. But the conundrum that scientists were faced when observing the cichlid fish and the finches is that after their points of divergence, so after these fish had established themselves as their own separate species, their own little groups, they seem to be evolving similar features separately. So they were spontaneously coming up with the same mutations, but that mutation would be required to have been present in multiple genes to have that similar feature. So there was a group of cichlid fish that formed this oddly shaped jaw, and another group at the same time of the cichlid fish that evolved this very odd shaped jaw, but that it was separate. It was an independent evolution. So the idea is, oh, wow, that these fish have developed these 20 mutations that would be required to have um, these similar jaw formations, and they've done that independently. But that wasn't the case. The explanation is that there was a single mutation of this BMP4 gene, uh, and a similar sort of explanation for why separate finch groups had evolved similar short, robust jaws, beaks for finch. So the expression of BMP4 leads to these developing these different jaw shapes. It boils down to that a higher amount of BMP4 in cichlid fish will give a shorter and more robust jaw, which is circled here. And for the Galapagos finch, a higher amount of BMP4 gives us a wider and deeper beak. The textbook does talk about a second master regulatory gene, CAM, which controls the length of the beak, but always remember, go back to your key knowledge, and all you are required to know about is the example of BMP4. So good to know about other examples just for understanding purposes, but BMP4 is the one that you definitely want to have a handle on. Okay, I think that is all from me, so enjoy and ask any questions you need to online. See you guys.